So, just picking up right where we left off in class, we talked about some of the biological functions of sleep. For example, we talked about sleep being an energy conserving strategy because it allows animals and people to rest and conserve their energy. That's time where you're not going out and foraging for food. You're able to um, have a period of reduced muscular tension, your heart rate, your blood pressure, temperature, rate of respiration are all at a calmer level. And so this is a way for your body to conserve energy because you're not physically doing things. Looking at sleep as a restorative process, these are all some of those biological functions and the reasons why we think we sleep. So if we look at it in terms of a restorative process, of course, we're able to replenish different kind of metabolic requirements. For example, proteins. We can rebuild and release growth hormone, for example, which we see released during slow wave sleep, which is that stage 3, 4 non-REM sleep. We know that it's restorative in the sense that when you are sick, it does help you heal faster, and then also it helps prevent you from becoming sick. So it helps you resist illness by building your immune system. And we've talked about this before, especially in relation to Alzheimer's. This process during sleep, this process of your glial cells flushing those waste products, this occurs during sleep in that lymphatic system to allow for your brain to get rid of those waste products. And we really see that primarily during sleep as opposed to when you're awake. So we've talked about the ties with Alzheimer's and how Alzheimer's might be linked to an actual sleep condition because you're not able to allow your brain to flush out those waste products as efficiently if you're getting shorter sleep, not giving yourself the opportunity to do that. And so some hypotheses and ways that we can kind of think about this. So, we know that your cells need energy, right? You know that there are particular events and functions that your brain has during the day that likely takes up a lot of energy. And if we're able to reduce some of this process, then we can replenish this and allow the cells to rest and replenish and restore some of these processes during sleep. And we also there's also an interesting hypothesis that fatigue and alertness may simply just be part of the circadian rhythm, and not necessarily to do with the wear and tear of the body, but just one of the process that we see as circadian processes where we saw that that rest cycle, that Brax cycle that we talked about earlier. So it's really interesting to think about fatigue and alertness simply being on a cycle, not necessarily just because of wear and tear. And again, looking at how we do very specific kinds of sleep deprivation studies, and again, how is it that we essentially recover from those sleep deprivation studies? Even though there's been a lot of work on sleep deprivation. It's a really, really very popular area of sleep research. Even though there's been a lot of work done on humans, there doesn't seem to be as many physiological effects, even in more severe sleep deprivation. Now, not saying you should deprive yourself of sleep. We do know over a period of time, short sleepers, people who aren't getting, 7 to 8 hours of sleep, that might be get severely less, like 4 or 5, they tend to be linked to a lot of issues, right? So we see a lot of comorbidities, and we also see people die younger. So it's not to say that you should continue depriving yourself of sleep, but in the short sleep deprivation studies where they might deprive you 1 or 2 nights, there's not as much physiological effect that we originally thought. Now, this 18-day mark, this actually comes from a sleep deprivation study that was done several years ago and actually hit the Guinness Book of World Records. And the Guinness Book of World Records has closed this down because they've deemed this too dangerous to do. The person who broke this record was a high school student. And he was doing it for a class project, and then it made the news, and then William Dement decided to get involved and to go ahead and monitor him. And so, he was a high school student, he was a teenager, he didn't seem to really feel the effects as bad. as you would expect. A lot of people say, oh, if you don't sleep, you die. And we know with fatal familial insomnia that there are some links that sleep is important to sustaining a healthy life. But with fatal familial insomnia, it's other brains degrading, or atrophying. It's not just the lack of sleep that ends up killing a person. We know that with animal studies, the only animal study that we can actually replicate where the animal does die from sleep deprivation is using rats and using a very specific method called disc over water. And if you use any other method of depriving sleep, it doesn't have the same effect. Whereas in rats, we see that it does eventually lead to death. It leads to a pattern discoloration in their coat, leads to them developing sores and just really not doing well. But in other animals, using disc over water, we can't replicate this. And using other methods with rats, we can't replicate it. So even though depriving you of sleep is not necessarily a great thing, it's not as deadly as I think that we originally thought it was. Additionally, the study before this particular one where it was the 18 days, there was a DJ that was doing it as a kind of like a stunt, and he was able to get that record in quite a few days, but not as long as 18. And he was using drugs, hallucinogenic drugs, all sorts of drugs to keep him awake. And so that kind of fueled a lot of what we assumed about, oh, if you stay up too late, you'll start hallucinating and all these things. But the guy that set this record here is a much better situation. It was more controlled, although it wasn't perfect. He got really cranky during the hours of the night that he really wanted to sleep, and he would almost beg them, "Can I just please go to sleep?" And they just kind of kept him up. And once he kind of each part of the night, 3-4 a.m. hit that really that really hard to cross zone, he actually was more pleasant after that. He would get a little cranky, he would get a little bit more aggressive and short-tempered. But they found that generally he did pretty good. He probably was experiencing something called microsleeps, which is just sort of pauses where the brain sort of shuts off for a little bit. William Dement came back and said, okay, I think that we really are seeing something new here that we hadn't known before. So that could have been sort of his brain making himself sleep. Now, in this study here specifically, we did see, though, of course, that there was some decreased cognitive performance, especially when the person when he felt like that task was really boring. And we see this in other studies as well. When a person feels like, oh, I don't really want to pay attention, they really struggle with doing the task if they're sleep deprived. So think about it when you are, sitting in class and this is something that you really, really don't want to be paying attention to. You tend to struggle really hard with that. And then, we see a difference in terms of physical tasks. The ones that require a lot of really fine motor skills do tend to do more poorly, but generally it's not as bad as we originally thought. And of course, talking about those microsleeps, this of course was a confounding factor. It can be a confound when you're doing these different studies because it is sleep, and if you're trying to deprive somebody and they're actually getting these little mini sleeps in, then you're not really depriving them totally of sleep. And so you can actually do specifically REM deprivation studies. And so you cannot deprive somebody of stage 1 sleep because that's the transition stage. But once they transition and go into stage 2, you can wake them up. In stage 3, you can wake them up. When they go to REM, you can wake them up. So remember that we have two types of sleep. We have REM and non-REM. And then REM does have it's no stages, it's just its own thing. And then REM, non-REM sleep, we have basically 3 stages, 1, 2, and then 3/4. Sometimes we call it a 4-stage thing. The field's kind of moved away from that since the slow wave sleep is very difficult to distinguish 3 and 4. It looks very similar. But you can actually deprive somebody specifically from REM by having them fall asleep, and then as they enter in REM, you basically wake them up. And it turns out that when the person the next night gets to recover, they tend to enter REM very quickly. It is harder to continue to deprive somebody of REM sleep. The next time they fall asleep, they jump right into it really quickly. We also call this REM rebound, where that person tends to jump into REM more quickly, but also spend more time in REM the next time that they have the chance. We don't really see as many adverse effects of REM deprivation as we see with, for example, slow wave sleep and non-REM sleep. We also see that in some cases, some antidepressants really reduce REM really, really significantly, and it doesn't seem to have as much effect on the individuals taking this medication. We also see in some cases some individuals with brainstem damage where they actually have complete loss of REM, and again, it doesn't seem to affect them as much as we would expect that it should. And we also see this in the case of locked-in syndrome, where the person has sustained some lower brainstem damage, but actually still is aware of their environment, but just not able to communicate that. So they're basically in a coma, and they can hear and see what everything's going around them, but they can't actually communicate that they're awake. But we also do see that those individuals may have complete loss of REM as well. And again, it doesn't seem to affect them as much as you would expect for this particular type of sleep. Now, we know that sleep is important for memory consolidation, which is that concreting of memory. Sleep is important for declarative memory, so things that you can talk about. We call it, things that you can declare. This is what we call explicit memory. It's also, we find that when people are deprived of sleep, they're more likely to create false memories. It's not that they're lying, it's just that they don't remember this story or the situation as well, and so they just kind of fill in the gaps. We know that REM is likely important for those non-declarative memories, which surprises a lot of people, that this dream state is important for those memories where you're actually doing something, right? So we're talking about like these implicit memories where you're doing a task, where you're learning a new skill. These are memories that these non-declarative memories are things that you are learning but can't really talk about. People often think of this as like muscle memory. And so this type of memory tends to be consolidated during REM, whereas declarative memory or episodic memory tends to be more consolidated during non-REM. And what's really interesting is that when we see when we see studies of mice doing different kinds of tasks, like going through mazes and learning new patterns and things like that, we see that the dendritic spines actually do increase, and they're stronger after sleep. And we've seen nap studies of this as well. So, just kind of giving us some evidence that sleep is helpful for learning and memory. And those mice that were sleep deprived, we saw, of course, the opposite of this, which is basically those dendritic spines reducing, so leading to poorer memory and poorer connections. And specifically looking at mice studies and looking at how a rat or rodent studies, how a rat specifically here is looking for a particular location. So if we take a rat and put them in a maze, or put them in an open field, and you try to have them look for a particular thing, like for example food, you can sort of map the hippocampal cells to see where in the brain the animal's activation is happening to find this item, and in this case it's food. It turns out that if you map this activity that happens in the brain and look at it while the animal is awake and actively looking for the food, okay, and then look at the animal during sleep, okay, you can actually see that the pattern looks really similar. And so what this does is, this is during non-REM sleep. So what this is indicating is that this place cell, or this particular series of cells and patterns that fire when we're looking for something, we're actively navigating our environment. If we're doing that during the day and then at night we see the same pattern, this is telling us that during sleep, your brain is going through the same activity and going back over those memories and going back over that pattern during sleep as it did while you're awake. So you can imagine this mouse is essentially going back over, those place cells are firing in the same pattern as it did while it's asleep as it does while it's awake. And so that tells us that that the brain of the rat is probably looking to see what memories it wants to keep and what it wants to go over, what it wants to get rid of. And that we actually have quite a bit of evidence for this in humans as well. And so looking at how a person has particular activation during a task while they're awake, right? And then during the night, the same activation pattern that we saw earlier in the day. And so in this case, this is during REM. So this was during non-REM. Okay, so these are place cells during non-REM. And then this is a reaction time task during REM sleep. Essentially, the idea is that these participants were dreaming of the task that they were doing, and that is going to solidify and organize those memories and strengthen those memories. So if you think about you're studying for a test and you're trying to understand this material, if you deprive yourself of sleep, so if you go an all-nighter, you deprive yourself of this process of essentially going back over those memories and concreting them and solidifying them. So you actually do yourself a disservice if you're studying and you pull an all-nighter, because you're going to remember more if you just give yourself the opportunity to do this consolidation process while you sleep. And there's a lot of other evidence for this as well. So this is a PET scan study, but there's some really good actual studies where you put electrodes directly on the brain. These are individuals, for example, who are undergoing surgery for seizures or epilepsy. And there's been some really, really cool studies looking at the same activation happening during sleep as a previous activation the day that day earlier doing the same task. And again, just growing evidence that these neuronal activity patterns are demonstrating the same thing, right? So while you're sleeping, you're seeing the same pattern that you see when the person was awake. And in this particular example, this was using a very specific kind of odor to try to kind of cue and prompt that memory. And so you can actually see that the brain activation during wake as well as during having exposed to this particular odor while asleep can help to kind of cue that memory to that particular task the previous day. So there's some really, really interesting studies and really clever studies to try to look at this activation and reactivation patterns that we see in awake as well as asleep. Now, of course, something that sort of challenges all of our hypotheses about how people sleep and the function of sleep is that there are some people that do get very, very small amounts of sleep. So they might only sleep 1 or 2 hours per night. And you might say, well, that just doesn't make sense for all the things that we know about sleep, if it's restorative, if it's energy conserving, if it's consolidating memory. But what's really interesting is that these individuals tend to be all-day nappers. So they might nap several times during the day. So if you look at a 24-hour span, they're not really getting just 2 hours of sleep. They're filling it in with a lot of brief naps. There's been a few famous people that have done this. Like Albert Einstein, for example, was supposed to be a napper and didn't sleep that much at night. And so, but if you take into account their sleep throughout the whole day, we do see patterns of more sleep than they're sort of leading on. Additionally, it might just be that some individuals just are more efficient sleepers, and so they don't need to transition with the stage 1 sleep. They don't need the less depth stage 2 sleep compared to the average person. They just might just be more efficient at what they do. Now, if we're looking at what's happening in the brain during sleep, we know that the forebrain is very, very important. So these are some of the systems. These are 4 of the systems that can that have a function when it comes to the mechanisms behind sleep. So the first one is this forebrain. So the forebrain system can display slow wave sleep independently, and we're going to talk a little bit about that in just a few minutes. And so the brainstem activates the forebrain into wakefulness. Okay, so the forebrain alone can display slow wave sleep. Then the brainstem is going to activate the forebrain to wake it back up. We have a pontine system, and that's going to trigger REM sleep. And then finally, the fourth system, we have a hypothalamic system. So the hypothalamus is going to affect the forebrain, the brainstem, and the pontine system to get it to essentially transition into sleep and wake and cycle through REM and non-REM. Now, if we look at some medications that can cause an unconsciousness, there are general anesthetics. Like if you're going to surgery, for example, your physician, your anesthesiologist is going to give you medication to put you into an unconscious state, right? So during this state, we actually do see similar waves, EEG waves, similar to slow wave sleep. It's not the same though. It's not sleep. Okay, it is similar. It does resemble slow wave sleep. Now, almost all of the general anesthetics that we look at, so if you look at these different ones here, almost all of them are agonists of GABA A receptors. Now, remember we talked about GABA A receptors before. It's an ionotropic receptor, and it allows chloride in. And chloride, remember, is a negative ion, and so that's going to hyperpolarize the cell and mean it's going to act as an inhibitory fashion. It's going to make the cell less likely to fire, and that's GABA, right? GABA is primarily an inhibitory neurotransmitter. So because so many of these medications affect GABA and mimic sleep, this makes us believe that perhaps it's GABA that helps promote slow wave sleep. So the brain system is using GABA in some way to promote slow wave sleep. And if you look at these agonists here, right? So almost all of these are agonist for GABA. And you can see that, there's minor agonists, some of them for glycine, some of them for serotonin. There's some antagonists for AMPA, for example, a little bit for NMDA, looking at glutamate specifically, but really they all share in common is that relationship with GABA. And so it kind of tells us that it's likely what's happening is that this particular neurotransmitter is important at least for promotion of slow wave sleep. Another really important thing that we have to think about is what areas of the brain are important, what systems are important for the different types and stages of sleep and getting us from sleep to wake. One particular experiment you can do is called creating an isolated brain. So this particular example is this is a rodent brain. If you make an incision here, this is what you would essentially do is cut between the medulla and the spinal cord. So here's the medulla right here and the spinal cord right here. If you make an incision here, you're creating an isolated brain. And when you do this particular experiment, okay, what animals do is they essentially show signs of sleep and wakefulness. And so even when you sever this part of the brain, you're still seeing the animal transition through sleep and wake. So that tells us that it happens right here, that this system right here is important for the transition of sleep and wake, right? Because you're severing this system here and it's not affecting sleep and wake. When you cut a little bit higher up, so if you do an isolated forebrain, so if you make an incision here in the midbrain of a rodent brain, what you see is that the electrical activity in the forebrain, okay, in the isolated forebrain, showed constant slow wave sleep, but no REM. So no REM. So that tells us that this area must be responsible for REM, and this area must be responsible for slow wave sleep. Okay, so the forebrain alone by itself can absolutely generate slow wave sleep, but it can't generate REM. Okay, and it's also not able to transition from awake to sleep. So it's important that this particular area must be important for slow wave sleep and has some mechanism, the system has a mechanism to generate by itself, but not REM. So we know that the basal forebrain area, which is right here, okay, is important for generating slow wave sleep just from further studies. And we also know that when we fall asleep, okay, and release GABA, that neurons in the basal forebrain become active, likely through this process of suppressing the tuberomammillary nucleus. Okay, so GABA suppresses the activity in the tuberomammillary nucleus. That's likely what is happening here, which also involves, if you're also looking at the different structures, the reticular formation, which happens further down here. Okay, this is what we see activates the cortex back to wake. So when you stimulate the reticular formation, and the reticular formation is not a structure, but just a series of neurons along the brainstem. When you stimulate this area, what happens is that if the animal's asleep, it'll wake them up. And if you have a lesion in this area, it will stop the brain from waking up. So this must be a switch, right? And it seems to be that the forebrain and reticular formation are guiding this transition between slow wave sleep and wakefulness. So the reticular formation works with the basal forebrain to guide the brain between slow wave sleep and wakefulness. And if you take my sleep class, we'll learn a whole lot more about the reticular formation and how this process actually works. And so we won't go into depth in this right now, but just know that if you take a sleep class with me, then we'll talk much more about this. Now, if you look at the pons, okay, if you look at lesions in the pons, you can actually get rid or abolish REM sleep. Okay, so remember the pons are here. If you do this, then you can abolish REM sleep. So clearly this area, and this is like the reticular formation down here, if you abolish, dissect out the pons or damage the pons in any way, you can actually get rid of REM sleep. So that tells us that the pons are important for REM. The subcorrelus is also important because we see that the neurons in the subcorrelus are active during REM, and what they're doing is they're inhibiting the motor neurons from firing so that you become completely paralyzed during sleep, and if you during REM sleep. And that's good because if you have tone, if you have access to your muscles, then what you're going to do is you're going to probably act your dreams out. So it's really important to have this intact, right? And so if you lesion the subcorrelus area, and that means that it's not able to inhibit motor activity, then you're going to see an animal, for example, act their dreams out. And this is exactly what we see when we have cats who have these subcorrelus lesions. They seem to act their dreams out. They're asleep. They're fully asleep. They walk around. They might meow. They might sniff stuff. They look like they're batting at something that isn't there. They're essentially acting their dreams out. And the idea behind a particular condition called REM behavior or REM behavior sleep disorder. This is a particular disorder in people, for example, with brain damage, maybe like a traumatic brain injury, Parkinson's disease, Alzheimer's. If they have damage to a very particular part of their brain, like the subcorrelus, they actually will act their dreams out. It's a very serious condition because these individuals, it can be really dangerous for them. And their bed partners, because they might fall out of bed, they might have a really scary dream, and that might wake them up. That might be a nightmare that wakes them up, and they essentially may hurt themselves, and so or hurt their bed partner. So it's a very dangerous condition to have. But usually we see this happen in areas like of the subcorrelus is essentially atrophied or damaged due to disease. So we'll pick up on narcolepsy on the next lecture, and so jump on